All right. Well, good morning, everyone. My name is Sara Suliman. I'm an instructor of medicine at the Brigham and Women's Hospital. And for the Discover Brigham demonstration today, I would like to present some of my work on identifying biomarkers to predict tuberculosis disease and how we can translate that discovery process into point of care diagnostics. So just by way of introduction, since we don't talk about tuberculosis a lot in North America, the global burden of tuberculosis, as you can see on this map, is still quite high. And before COVID, it was actually the highest, it had the highest level of mortality from an infectious disease globally, according to the World Health Organization. You can see that there's a high concentration in sub-Saharan Africa and South Asia, and in some parts of South America as well. So the daily mortality rates actually are on par between tuberculosis and COVID-19 these days. There are about 4,000 case fatalities a day. Just to orient you, both are respiratory diseases. I'm focusing on tuberculosis for the purpose of this talk. And it's characterized, the main form of tuberculosis that is presented in adults and adolescents is pulmonary tuberculosis. And it's usually characterized by these white nodules on X-ray radiographs, as you can see here. There are different transition states during the process of TB development. Not everybody who gets infected with the bacteria that causes TB, Mycobacterium tuberculosis, actually progresses to active TB or clinical TB disease. The estimate is actually that 5 to 10% only of those who have been exposed to the bacteria would actually develop active TB disease. And of course, the second transition point would be from diagnosis of active TB to initiating treatment and predicting different treatment outcome. So we need biomarkers to predict who will transition into these different stages, who will successfully complete treatment. There's a lot of work that I have been involved in, and also just globally on identifying biomarkers that predict this transition from infection to active disease. And then from active disease, having biomarkers to confirm the diagnosis of the cases. We still use very old diagnostics for TB. So having additional biomarkers to confirm the diagnosis, especially if there's a differential diagnosis of something that looks like TB. And finally, we need biomarkers also to predict the likelihood of treatment success. So whether after treatment you will be reinfected again with Mycobacterium tuberculosis, whether you would relapse, or whether you would have a sterilizing cure, meaning that you get rid of all the bacteria and go back to a healthy state. So the biomarkers that I'm describing in this presentation are host biomarkers. So this is referring to gene expression signatures in the human, as opposed to signatures that are associated with the bacteria, which makes them a lot more accessible and unique in that case. So of course you can think of host blood biomarkers as non-specific to the bug, but actually what has been clear is that you can identify specific changes in the genes that are expressed in the human that would reflect specific infections, be it viral infections or bacterial infections. So this concept started about 10 years ago with the seminal paper in Nature. And what they did is they took whole blood samples from many individuals. There were actually two cohorts, one from the UK and one from South Africa. And you can see that when they combine individuals who are healthy, they have no evidence of clinical TB disease, and individuals with active TB disease, and then analyze whole blood gene expression on this microarray chip. So this is a chip that reads how much of each of these genes is expressed in these individuals. They identified 393 genes that were uniquely different. One group was higher in people with active TB that you can see in red, and one group was lower. So this was the first indication that you can actually use host blood transcriptional biomarkers to differentiate between people who have the disease and people who don't. And what was interesting also in this study is if you use the score that they developed actually from these 393 genes, you can start to see that there's a clear correlation between the score and the extent of disease. So if you look at the lung images that I showed initially, you can see that there's a clear increase the higher the radiology score is. So if you go from no disease, so no evidence of white nodules on X-ray, to minimal disease to moderate disease, you can see that the molecular score developed from these genes would increase as a result. And since then, this has been a very active area of research. So there's been work on diagnostic signatures. So at the time that you have the disease, what are the markers that differentiate that? And longitudinal biomarkers that predict risk of progression to TB disease. So if I'm healthy now, what's the likelihood that I will develop TB in the next months to years based on the changes in my blood gene expression? So one important point about TB is that even though the infections are quite chronic and take a long time from the time that you're exposed to Mycobacterium tuberculosis to developing TB, the highest rate of progression to disease actually happens within 2 years of infection. 2 years is still a long time. The incubation period for a lot of viral infections is in the order of, with the exception of HIV, it's usually in the orders of days, maybe weeks. But in TB, you can be infected for a number of years before developing TB. However, the majority happen actually within the first 2 years after infection. So this is a nice window to focus on trying to develop biomarkers to predict within 2 years of infection whether or not you will be progressing to active disease. So the first study that I will talk about is called the Adolescent Cohort Study. So this is based on adolescents in a town called Worcester near Cape Town in South Africa, not to be confused with Worcester, Massachusetts. From a large cohort of 6,000 individuals, the study identified 46 individuals who developed active TB within a span of 2 years from enrollment in the study. So at the time of enrollment, as people probably know, you can use either a skin test or a blood test to determine whether you've been infected. The positivity on these tests doesn't mean that you have disease. It just means that you've been exposed to Mycobacterium tuberculosis. So it is still possible that there has been an infection episode before enrollment in the study, but this is the best evidence we can have to confirm that at the time of enrollment, they were infected. And they were followed up for 2 years. And then, as you can see here, people progressed to TB within the 46 individuals who developed TB disease. They progressed to TB at different time points. So some of them were closer to 6 months. Some of them were more were closer to the 2-year follow-up period. So what we did then is we realigned everyone to the time of diagnosis so that you can actually go backwards and use their blood samples that have been collected every 6 months to discover what gene expression patterns have been different prior to development of disease. So we're not interested at the time of disease. We're interested in individuals who appear healthy but are on the path to becoming sick. What's the process for developing a biomarker from these samples? So we just collected blood samples in these PAX gene tubes. So they're, you just basically input 2.5 mLs of blood into this tube that has a lysis buffer. You can freeze it for years and then decide when you would retrieve it for analysis, which is very useful for large field studies. And then you can choose what samples to actually analyze as opposed to analyzing thousands of samples just to get a signal from a few individuals. And then we extracted RNA samples from these blood tubes and subjected them to RNA sequencing. So for those who are not familiar with it, this is a process where you actually sequence each individual transcript that's generated from a gene. and quantify how much of that gene is expressed per sample. And one important point is that for each gene, there are these regions that are called exons. So those are the expressed parts of the genes, and introns. And you're more interested in exons because we want to translate this into a point of care platform, so we can use other techniques like polymerase chain reactions or PCR to develop a point of care tests. So we want to focus on the areas that are actually expressed, and also on the junctions, so that you prioritize detecting the mRNA as opposed to detecting genomic DNA. So we quantified these exon-exon junctions, and then from that, we applied a machine learning algorithm called paired support vector machine, which basically identifies a line that best discriminates between cases and controls based on the expression of that gene. So it's paired because we're using the expression of two different genes, and we do this iteratively over multiple genes that best discriminate between cases and controls, and find the line that defines at which point you're more likely to become a case than a control. And we try to we did actually down select the most predictive genes and analyze those by PCR, and then validated the performance of the biomarker on test set samples. So those are samples from the same cohort that have not been included in the discovery process. And this is a very important part in machine learning and biomarker development, is that you want to test your algorithm or point of care test on samples that have not been included in the initial set to validate the performance. And as an additional rigorous method, enrolling people in a completely different study, and even better if they're from a completely different genetic background and location as well. In order to validate the performance of biomarkers that we developed from those South African adolescents, we actually had access to samples from a completely different study, which is adults who are household contacts of active TB cases. So we had samples from Ethiopia. This is the site called ARI, from the Gambia in West Africa, and also from South Africa. We had some samples from Uganda, but not enough to include in this study. So based on the sample numbers, we also included training samples from both South Africa and the Gambia. Training meaning that these are samples that we can include to discover new biomarkers in addition to the ones that we discovered in the previous study. We perform a blind discovery on the genes that best discriminate, and then validate the performance both on test set samples from South Africa and the Gambia, so samples not included in the discovery, and then on an independent cohort on Ethiopia. And then now that we have access to both studies, we can use the discovery from the adolescent study to validate on the household contact study, and vice versa. We can also use the household contact study to independently validate it on the adolescent study. So similarly to the adolescent study, the household contact study is called GC6, which is just Grand Challenges 6, funded by the Gates Foundation. Very similarly, you enroll individuals, this time based on a known exposure time to someone in the household who has active TB. So the main difference between the two studies is that here we have a definitive time point that we know someone has been exposed to Mycobacterium tuberculosis. It doesn't preclude that, you know, they may have been exposed before the time that they started living with someone with active TB, but at least we have a better epidemiological time point to say, okay, I have some confidence that at least during this period, this is when they had exposure, be it primary or secondary to MTB. And very similarly, we followed them up for 2 years, and now we can go backwards and analyze the samples at the time that the individuals were healthy before developing TB, and use those to predict risk of progression. So one important concept when you want to measure the accuracy of TB biomarkers is how to measure the sensitivity and specificity of the test. And you might be hearing those terms a lot, especially with COVID diagnostics now. We use this confusion matrix, which I find the name quite accurate, which basically compares the performance of your actual biomarker to the real gold standard state. If you have a definitive clinical diagnosis that someone has TB or doesn't TB, use that as the gold standard. And usually people use mycobacterial culture to make sure that you actually have bacteria in the individual that you suspect to have TB. And then you compare the performance of the test that you developed. So sensitivity is telling you how many cases, how many positive individuals are detected accurately. And specificity tells you how many of the people who are uninfected, or in this particular case, who did not develop active TB, are detected accurately. You can flip it, so you look at the precision or the positive predictive value. So that tells you, out of the individuals that are called positive by my test, how many were actually cases. And the negative predictive value is how many of the individuals who are called negative by my test were actually non-progressors in this particular scenario. And accuracy just combines both the specificity and sensitivity. So overall, how well does my test perform to call a case or a control accurately? And one way that I'll be showing in the subsequent slides to show the accuracy of the test is to draw this receiver operating characteristic curve. So this ROC curve shows the dynamic range of the values that you get and how well they correlate with the disease status. So if you have a perfect square, when you plot the false positive rate and the true positive rate, that means you have a perfect classifier. My test always predicts progressors and non-progressors accurately. This is an ideal scenario, which doesn't usually happen. And then if you have a random classifier, you're usually in the range of this diagonal line. And what you want is to have at least some significant difference between the line that you get based on the performance of your test and the random classifier. From the adolescent study, now we started applying this to samples from the test set first. So sorry, from the training set. So just to test within or cross-validate within the same samples that were used to discover the test, did they actually predict TB? So you can see that from 6 months before developing TB, or 12 months, or 18 months, you get a significant prediction of risk of developing TB. But when you go further than a year and a half, then you start to get in the range of random prediction, meaning that your test doesn't do better than flipping a coin, basically. Now, remember I told you that we started with a full RNA sequencing, meaning that we sequenced every gene in the cases and controls that were enrolled in the study. So this is obviously a very expensive process and not very easily translatable into point-of-care diagnostics. So we actually developed a PCR test that would bypass having to sequence the entire genome. And we can see that based on selecting the best performing genes, the performance of sequencing everything versus only using the samples, using the genes that best predict TB, we get this significant prediction, which comes to about 70% accuracy. Before that, we did not have any way of predicting whether someone would actually develop TB. Even though 70% doesn't sound impressive, this is a huge improvement from the state of the field before. And now, as I mentioned, we had samples from other sites. So this is South African samples that were from a different cohort, not from the ones where we recruited the adolescents, and the Gambian samples. You can see that even though the test was developed in a completely different population, different age group, it still predicted progression to TB. And not surprisingly, based on this result, we can see that we predict TB better 1 year before developing disease as opposed to 2 years. So one interesting thing is that when you use that biomarker as a score and then go back in the samples to see how well, how much does that signal change longitudinally as you approach developing active TB disease, you can see that the controls have basically a non-existent score, as you can see in the black marks. And then as the score gets redder, that means you have a higher expression of those genes that are associated with developing TB. So you can see that even as far as 1.5 to 2 years, you start to see that there is a signal in individuals who will develop TB. It's not very bright, as you can see at the bottom, but it starts to get more red the closer you approach TB. So that is consistent with the idea that TB, first of all, is a very chronic disease. It takes a long time to develop. And the immune system starts to change as far back as 1.5 or 2 years before you actually get full-blown bacterial pneumonia that's associated with the TB disease. Yeah, so now to the cohort that I just described, which is the household contacts. As I mentioned, we had samples from South Africa, the Gambia, and Ethiopia. This was a huge consortium of researchers, both from Europe, North America, and Africa. And I had the pleasure of working with the consortium as a postdoc in South Africa. So the main question, which is more practical in terms of developing public health interventions is when I live with someone who has TB, will I develop TB myself? What's the risk of me developing the active disease? I might be infected, but as I mentioned, only 5 to 10% individuals actually move from the infection to the disease. So how do we refine the prediction of whether someone will get sick? So maybe I can put them on antibiotics before they get into full-blown active TB disease. And we performed exactly the same approach that we did for the other study. And in this case, our goal was to reduce the number of classifying genes to as few as possible. And we managed to down select 4 genes. So no control genes, just 4 genes, and then looking at the ratio of the expression of 2 genes that are up in people who would progress to disease and 2 genes that are down. And we can form a score based on the ratios of the expression of the genes that are up to the genes that are down. So because it's 2 by 2, we have 4 different scores that we can average and use as an algorithm to predict TB disease. So a much easier test to translate into a point of care platform. As you can see, in the individuals who didn't progress to TB disease, the score is 0. And as far back as 2 years of 2 years before developing TB, you start to see a very clear enrichment in this pink color, which indicates that you have a higher score for developing TB disease. And these are just plots to show the accuracy in different scenarios. So if you look at the entire data set, it's about 0.67, which is 67%. The best prediction happened in South Africa, but then we also got a significant prediction in samples from the Gambia and from Ethiopia, which is interesting because they're different populations. They have different circulating MTB strains. So this shows that there is a universal signature that predicts whether someone will develop disease. And different from the other study, we found that whether you go 1 year back or 2 years back, you still get similar prediction. And we think this is because of the exposure to the household contact. At that point, you get an acute exposure to someone who's shedding a lot of bacteria, and that causes inflammation and sets the cascade into progression. Whereas in the other study, as I mentioned, you're starting with individuals who are infected at the time of enrollment, but they could have been infected many months before that, or they may have been exposed again after. So here we have a clear exposure time point, and that makes the test more accurate in predicting disease up to 2 years. And if we just look at the samples that are collected when you were at the time of contacting someone who has TB, we have about 70% prediction. And as I mentioned, we also validated on a different data set. So we went back to the other study and used it as a validation cohort, and we also got 70% accuracy of the test. So 70% seems to be the ceiling that we can't break. And that might be because there are other different pathways to developing TB that rely on different sets of genes. We just did not have enough individuals to start to develop that even more. So moving on, we wanted to, first of all, generate the best predictor for progression to active TB disease, but also move forward with trying to translate that into a point of care test that relies on something as simple as a finger prick and a small volume of blood. So we have these we combined the two studies and then identified what are the best predictors. And we found this set of 6 different genes. So only 6 genes, 3 that are up in people who might develop TB, as you can see in red here, and 3 that are down. So you can see if you look at how the genes are expressed during the time course of developing TB, these are the genes that go up, and these are the genes that go down. So now we can use those again to develop ratios to generate a score. So it's a very straightforward test and doesn't require normalizing it, for example, to control genes or anything of that sort. And we looked at the accuracy of just the 6 genes compared to the initial model, which had 16 genes, and actually multiple different transcripts within that gene. And you can see that the accuracy is very high. Even though it's slightly higher with the full model, it's actually the difference between the two tests is not statistically meaningful. So what does that mean? So the World Health Organization has criteria on the minimum performance that, that you need to actually push a test forward to marketing. The minimum criteria for what they call target product profiles, which you can find on the WHO website, are that you should have a 75 specificity. So specificity is detecting people who are not at risk of developing the disease, and 75% sensitivity, so detecting those who are at risk. Optimally, you obviously want more than 90% for both. So what we can see from the risk 6 score is that we actually meet the minimum criteria, but we're not close to the optimal criteria. And if you go back more than a year, then you fall off the minimum score for the WHO requirements for a target product profile. So these tests seem to have a limit in terms of how far back you can go, so you can reliably detect someone's risk of developing disease. And in terms of now going to a different cohort, unfortunately, the performance of the test start to drop. It's still better than random, but it's not meeting the WHO criteria. So there's still a lot of work to be done, maybe in terms of combining this test with another test, or using this test as a triage, so you can use another confirmatory test based on a different sample type, like saliva, for example. Or there's work being done on urine samples to look at TB shed molecules there, and whether combining, looking at whether combining blood gene expression biomarkers with another platform would improve prediction of TB disease is now an area of active research. So one important application also for biomarkers is if someone comes with clinical symptoms that look like they are TB, someone is coughing, they're losing weight, is that TB, or could it be something else? And this is becoming very important now because the symptoms of TB and COVID are very similar. You know, you have a dry cough, weight loss. So if they both have what looks like pneumonia, is that caused by MTB infection, or is it caused by a viral infection? So we did an analysis where we actually compared the performance of TB. So definite TB or probable TB, these are based on how much mycobacteria are actually able to culture from the sputum of individuals who have the disease, and compared those to other respiratory diseases. So knowing that someone has a different viral infection, for example. We did find that our biomarker works better if you have a definite TB. But there's still a little bit of overlap between the two, so it's not entirely accurate. So that means we do have a way to prioritize individuals who are likely to be TB patients, but we do need another confirmatory test. And as you can see here, for diagnosis, you need the WHO requirements are even more strict. So you need at least 90% as a minimum criteria and 95% as an optimal criteria. And to distinguish TB from other diseases, we don't meet that strict threshold, but we do better than random. So now the question is, well, if I put a TB patient on treatment, how do those biomarkers perform? And now if you're comparing people who have TB to those who are cured from TB, you actually get a really good prediction. So meaning that the blood disease score correlates with active disease at the time of disease, and then that resolves with treatment. So it's likely because of the high bacterial burden that you have at the time of disease that that results in the changes in gene expression that you see in the blood biomarker score. So now you can see that the TB treatment is really long at 6 months of antibiotics. And the majority of change actually happens really early on in the treatment. And then by the end of treatment, end of treatment, you can see that there is a significant reduction from the beginning. It doesn't get to the level of the healthy controls, but if you follow up these individuals further, eventually they do return back to baseline, meaning that they resolve the inflammation associated with TB. And because both studies that I described earlier were based on African populations, how universal is the signature? So we had access to samples from both Peru and Brazil, so South American populations, where they have, again, a very different genetic background, different circulating MTB strains. And we see that there is still a significant prediction based on based on the biomarker that we developed in African population. So you can see that there's about 90% prediction in the Peruvian samples, and similarly 91% prediction in the Brazilian samples. So back to the main point that I mentioned earlier, which is, can we translate these biomarkers into point of care tests? It's not really feasible to collect a sample, extract RNA, and run a PCR before you get a test, especially if you're in the field and you want to screen individuals at different households. So we're trying to develop a finger prick test that requires minimal involvement, and ideally you want the individual to be able to do it themselves with minimal involvement of healthcare providers. We are not there yet, but the work is happening. So we compared the performance of the 2.5 ml, which is what we used in the PAX genes I described earlier, versus a finger prick that takes 50 microliters or even 20 microliters. And you can see that the performance of the test is still significant and not meaningfully different from the one where we developed, where we used samples with the full amount of blood for extracting RNA. So with that, I have a few concluding remarks. We can actually define host blood biomarkers that define risk of prediction, prediction of risk to active tuberculosis. And it transcends both population and geographic heterogeneity, which means that the way the immune system reacts to the MTB infection and whether or not you develop active TB is a universal mechanism. And both risk 4 and risk 6 are simple blood biomarkers. And we're in the process of trying to translate those into point of care testing. And the main application, which actually is happening right now, is to enroll these individuals into clinical trials to see if I predict that someone is likely to develop TB within a year, for example, can I put them on preventative therapies like host-directed therapies or antibiotics to prevent their development of disease? And the early trial results are actually very encouraging and suggest that if you go back 6 to 9 months and put these individuals on preventative antibiotics, there is a significant reduction of their development of TB disease, which is really encouraging for the application of these point of care tests. So with that, this was a huge team effort to enroll all these people in different sites. This is the consortium that I was working on when I was in Cape Town. I would like to acknowledge everyone and my colleagues in Cape Town and Seattle who have been fundamental in carrying this work forward. Thank you.